Yeah. So we are in the week 2 of the introduction to the proteogenomics live session. So previous week we had seen the different methods of the DNA, how it is sequenced, and how the next generation sequencing has helped us to understand understand the sequences and what are the SNPs, how it can be identified. And now we are just linking both the proteomics and the genomics technologies for understanding of what is the effect of this SNPs at the proteome level. So here I would like to, before going into today's thing, so firstly I'd like to give some example of the recent published nature, that how recent published articles, that how these both the techniques have helped us to understand it much deeper way, and why it is much important in today's time to understand both the sets. So for example, this is they have shown the CNA and the mRNA, that is the copy number analysis, and the mRNA correlation, and the copy number analysis and the protein correlation. So here this graph shows that they are with the copy number abundance or the copy number variations in the genome level has an effect at the mRNA level, but doesn't always, and in the protein abundance it is not so. So though it is at the mRNA transcript level, there has been the effect of the copy number abundance, but in the protein it is not there, and it has a significant negative correlation, that though there is the copy number the abundance is available in the genome sequence, it is not reflecting in the protein. So these phenotype expressions are needed to be monitored at the genotype level to understand that actually what is happening at the phenotype level. So here you can see that it has mentioned that these identified 17 regions of the significant focal amplification and 28 of the significant focal dilation. They calculated the Spearman's correlation with both mRNA and the protein abundance. So the genomic technologies helped us in understanding the different uh, SNPs, different mutations, but what is actual reality in the phenotype level or the protein level, what is the actual thing happening. So there you can see that this correlation matrix did not always correlate, that the copy number abundance means will be reflected at the protein level. So another thing they had done was to further investigate further the cis effects of CNS, that is the local presence in the particular region, where so here you can see that the CNA mRNA correlations were same. So the test that is generally used for this kind of correlation finding is the Kolmogorov-Smirnov test, which is a kind of a statistical test to find out that whether the correlation is significant, is same, is higher, or it is lower. So here you can see that the CNA protein correlations, then the genes in the non-focal regions, it is not correlating. It's but in the, of course, it is much significantly correlated in the mRNA abundance. So these tests, all these results, all this shows us the need of understanding and utilizing both these technologies, or combining both these technologies to get the holistic picture at the molecular level alterations, how it is happening and what is the actual effects. So now in the proteogenomics approach, in these both techniques, one major role which is played in the gene transcription and the regulation is the epigenetics, or the role of epigenetics. So what is the epigenetics? So whatever things are happening in the sequences of the DNA sequences, but always it is not only the factor that gets transcribed and translated to the at the protein level. So major thing, that another thing which is beyond our genetic code is the epigenetics. So what is epigenetics? That is that which are the modifications that are regulating the on or off the gene, switching on or switching off. And so these are the modifications attached to DNA and not the sequence. So now you can appreciate the fact that knowing only the sequence doesn't make our case easier or doesn't lead us to understanding of the whole things easily. So here we know that the chromosome, the DNA is the sequences are packed in such a way with the different kinds of proteins so that it gets easily comes inside the nucleus, and when the packaging breaks up and breaks open, then the transcription factors and the different proteins can interact with the DNA sequences, which can lead you to the transcribe. There is a huge role of this epigenetics in understanding that how and these modifications, that how the DNA sequences will further translated at the protein level. So for this reason, to understand epigenetics is quite important. So then only uh, it will help us in understanding that why these correlations are not matching or how these correlations are working. Yeah, so for this first technique, what we will be looking at is the chromatin immunoprecipitation assays or the cheap assays. So what is this? So it helps us to understand the protein DNA interactions. It helps in understand the protein DNA interactions. So how it is done? So first thing is to done is that we cross-link to trap the protein DNA interactions. So we know that the number of proteins gets interacted with the DNA. There are histone proteins as well as some other kinds of proteins. So first we need to understand that how these protein DNA interactions are happening, or what are those regions of the DNA that are interacting with a particular set of protein. Okay, so this is for which is for
Hello.
chromatin. Yeah.
Yeah, Ankit, if I can just interrupt you, I think there's a question from Srishti, I think.
Yeah. Yeah.
Can you please
So
ask the question? Srishti, do you have any questions? Okay, I think Ankit, you can go ahead.
yeah. So if there is a question, you can ask. Otherwise, okay. Yeah, so whereas the chromatin immunoprecipitation is help us, so we know that particular set of proteins that are interacting to the DNA, but if we do not know that which part of the DNA is interacting, and we need to understand that what is that part of the DNA which is interacting, so we can do the chromatin immunoprecipitation assay. So how we do this? So we just perform cross-linking, so mostly using formaldehyde or to stabilize those cross-links, which to help to understand that the protein and the DNA interactions get stronger. Then what we do is that we perform the cell lysis. Why the cell lysis is done? So in this cross-linking, so we need to uh, bring out the nuclear proteins, as we are the cells, followed by the cytosolic proteins needs to be removed. Then we need to extract all the nuclear material, yielding unbound nuclear proteins. So where the DNA and the protein is not bound, full length chromatin and the cross-linked protein DNA complexes. So everything is extracted out. Now these extracted genomic DNA are shared into small pieces in the range of 200 to 700 base pairs. So this gives an optimum region for which after which you can perform the PCR and can identify the sequences. Then the immunoprecipitation is formed. As you know the exact amount kind of protein of which DNA region you want to isolate the fragment of that particular DNA, that protein is interacting with which part of the DNA. So you know the antibody, and you will do a immunoprecipitation. Where will you precipitate the proteins from that particular antibody? Then you will purify it, and followed by elution of the protein DNA complex, and then sequencing of the DNA and mapping. So this is the whole protocol. So but here there are three steps that we need to keep in mind. When first is the range of this DNA, and then how better, and the elution and the purification steps, which doesn't hinder in the further sequencing steps. So this is the one kind of assay. We use the chromatin immunoprecipitation assays. Now, so here is the sample preparation. This is the concept, and this is how the samples are prepared, and then this is sequence. But next step, the major step, vital step requires is the computational analysis or the data analysis, where to find out that which region of being mapped, that is that particular protein is getting attached to your particular DNA. So for this, we generally follow this kind of pipeline, where you map file normalization, then read the data, and then you will find out that this particular region, you will find peak, will call the peaks, that you call that region, and then you will annotate those, and then you will do a motif analysis or gene ontology. These are in the secondary part of that after you got the particular genomic regions, then you can try to find out that it is in the motif region, uh, or what is the role of those particular region of the DNA in that in the further analysis. So this is that how this computational analysis helps us in mapping the region of which gets attached to that particular DNA. But so now let us look at the real some data analysis software and the tools that are available, which through which people generally performs this kind of analysis after the sequencing has been done. So there are a number of chipsec data public data sets available. So for example, to download of the histone, some all those proteins of the histone modifications or some other uh, proteins. So this is one of the portal called the ENCODE portal, which is of the NHI, National Institute, NIH, National Institute of Health, which creates some of those chipsec data sets. Others are like roadmap epigenome database, IHEC data portal, epigenome database. So these from here also you can download and can try to analyze the data sets with the tools that are generally used. So this is the how encodes data you can try to find out, like in the it is the histone chipsec, so whatever modifications and the histone related proteins data is available here. So this is encode in the total encode you can try to find out. So how the data analysis is done? So the sequence reads. So after you perform the sequence, so after you have immunoprecipitated it and you have sequenced it, so the sequence reads in the FastQ format. So whatever sequencing generally comes is in the FastQ format, are mapped using tools such as your Bowtie or BWA. So these are the tools which helps in alignment. So it is exactly the same as we do for any other kind of whole genomic sequence data or whole exome sequence data. So we just try to map the sequences, align those sequences, and to find out the different that how what is the similarities and dissimilarities. Their objective remains different, and here objective remains different. To here it is done to find out that where it is matching, so that one can understand that which part of the genomic region it belongs. So this then it gives us the SAM and the BAM file. So it's a binary alignment map files, and then the after alignment, reads are filtered as redundant reads, and the remaining non-redundant reads are used for analysis. They're just if it is coming in the same genomic region, so those are filtered, and then it will be further used for analysis. So now after this, here what is different from your, if you do a whole genome sequence analysis or a whole exome sequence analysis, so here there you will be trying to find out the variants, right? So where is the my variance, or where is my particular positions or the polymorphisms in the particular DNA in the of a disease condition of a, or of a different condition from the normal. But here you will be doing a peak calling to identify the significantly enriched loci in the genome, so that because here our objective is different. Here our objective is to try to find out that where it is, from where region of that particular DNA the proteins are interacting. So here comes the difference in the analysis steps. So here peak calling results are here, like in the mostly uh, it is bait format is a format through which this is written. So do not have stand information from the gene information with the histone marks that are enriched around TSS. So this is around transcript start site. So mostly on the histone marks are enriched around your transcript start site. So MAX2 is one of the very frequently used calling tool. So to identify those peaks or to find out the significantly enriched loci in the genome. So there are also many tools, but all doesn't comes we each comes with a different pros and cons. So you need to understand that how it will perform. So this is the max model based analysis for chipset. So if you can try on your own, try to using the model based analysis of chipset from downloading the data and then aligning and then trying to find out that how it is. Second, and the third most important thing is that, so as I had previously mentioned also, that the quality check or the quality control is very much important for any kind of sequencing data. So here also the quality check is very much important or critical to judge uh, whether the data are of high quality and suitable for further. So these are the some of the parameters through which it is checked that the how the data is quality for the chip assay. So like in the normal sequencing data of the whole exome sequence or the whole genome sequence, there are different parameters like the Q30, the GC reads, the sequence duplication rate, etc., which we will try to look in our in the latest sketch today. So here we can see that like the mapping ratio, it reflects the read quality and the proportion of the sequences mapped, which is there's a sequence reads derived from the true genomic DNA. So here it is should be over 80%. So each parameter has how and what is the cutoff should be maintained for to understand that it is of high quality data. Then is the read depth. Sufficient depth, read depth depends on the and the antibody S by N ratio. S by N ratio, the ENCODE consortium suggests at least 10 million uniquely mapped reads as a minimum to analyze sharp mode peaks of the human. So these are the different parameters and their cutoff. So then is the library complexity. It ranges from 0 to 1, and the ENCODE. So ENCODE BS consortium, that is being generally regarded as of the standard and the highest quality, that the complexity of greater than 0.8 for 10 million mapped reads, and lower values indicate excessive PCR amplification or small amount of initial DNA. Second is the normalized strand coefficient, SN indicator for sharp and broad marks. So this also should be less than 2. Then background uniformity and the GC submit bias. So all this region, so all these parameters need to be checked before going on for the further analysis to understand that our data is of high quality. So this is how generally it is looks for each sample wise. So for example, these are the different modifications of the histones, and these are SS, these are the your parameters and how it.
Number abundance.
Sorry.
What is copy number abundance?
Copy number abundance is one where in the genome sequence, what happens is that sometimes a particular sequence, this your nucleotides are repeated certain number of times, but in certain conditions or in your diseased condition, it is repeated much in a much greater number than the what is actually present in the normal individuals or the individuals which doesn't have the disease. Okay.
Okay.
Then we have this background. So this through the correlation matrix. So whenever we do in an omics data set for say to identify or understand that how the samples are of the quality. So what we do is that we generally try to find out the correlation among those. So here we can see that there are some of the samples which shows a lesser correlation than the other, so which depicts us that it is not of the same or of the highest quality of the we along with the remaining samples. So by these parameters we can understand how it is getting changed. So then is the after we analyze the data, what we do is that the normal. So there are various reasons to normalize the data. One is to always to reduce the technical biases and the variances to understand whenever we try to compare. Now, if we compare between condition A and condition B, we should keep in mind that the comparison can only be done when all the both the sets are on the same parameter. So that's why normalization can be done for the peaks and all uh, for analyzing that okay, this condition and this condition, what is the difference in the loci regions of those particular DNAs in the epigenomics. Then final is the functional analysis, as I mentioned that the motifs. So if you know that the motifs and the sequences that whether these particular fragments of the DNAs are region of the motifs, and if the protein binds to it and it does not allow it to functional, or if it is vice versa or anything, so you can do the functional analysis and the motif analysis and the gene ontology analysis. So this is overall of the chromatin immunoprecipitation assay, which gives us to understand that how these particular proteins and are interacting with those DNA, and how we can understand that what are the roles if that are present of the particular motif or particular region, how those proteins are affected. And this is the overall after the sequencing, after we will follow the workflow to finally identify those particular regions of the DNA. So anyone has any question regarding it? Not right. Okay, so I guess everything is clear. So then one tool is there known as Droma Plus. So this is an overall chipsec pipeline tool that includes your quality check, analysis, and visualization of multiple chip samples. So you can uh, uh, try to use this tool to try to identify from the available publicly available data sets, and can try to find out that how it is coming. Okay, so this is one tool which you can use. Otherwise, all those tools in a separate publicly available, that BWA or Max 2, you can use those individual tools as well, and can try to find out that how it is uh, how different are the results, or for curiosity, or how same is the result. So you can perform that as well. So the here you can find out that how what map file formats can be used and how it provides your result. Yeah, so this was for the chip chipsec analysis for finding one kind of interactions in the field of your epigenetics or uh, epigenomics. So now is one other is the DNA-seq analysis. So here another thing is that we know that their nucleosomes remain nucleosome remains the in the DNA remains in the structure. Observe that whenever the nucleosome disrupted regions are there, then that region becomes accessible for all those transcription start sites and all those polymerases, and it helps in the translation of those particular regions of the protein. So to identify and to understand that particular part of the region of those genome, so this is the analysis which is being used is the DNase-seq analysis, which maps the DNase1 hypersensitive sites. So these sites are also sensitive to the DNase1 enzyme. Okay, so these sites are also uh, very much sensitive to the enzyme DNase1. So nucleosome disrupted regions are generally associated with the gene activation. Okay, so this is the rule. So this what happens is that when we provide the DNase1 sites, it captures across the whole genome where this site is available. So it cuts into those fragments, and then by high throughput, then we perform the sequence. The sequences is then the sequencing is performed to identify those particular regions of those DNA. So you can see that here first we will cut with the DNase1, and then we will try to ligate those, and then it is the sequencing is done. Hopefully it's clear, right? So this is how to identify those nucleosome disrupted regions and the most associated with the gene activation regions. So that DNase-seq analysis helps us to find out. So again, it's if we say that the analysis, so analysis is more or less again the same with raw DNase-seq data, then with the data pre-processing with adapter trimming and filtering and sorting. This is exactly the same what happens with other normal sequencing data analysis, quality control and read mapping, then DNase1 footprinting, that is after the peak calling. So peak calling is again the same to identify those regions of those from the whole genome and find significant cleavage depletion in peak regions. So this is exactly the same how this happens, the analysis for the DNase-seq, but here the principle or the objective is different from the ChIP-seq analysis. So here we are trying to find out the parts of those DNA where the most participation of the gene activation happens for the translation into a functional RNA or the protein, and the nucleosome disrupted regions. And so this is the these are some of those examples of the protocols of the DNase-seq. One is your end capture, identification of what whole genome scale, then DNase flash ones. These are just different kinds of experimental protocols that you can go and see that what are how why these are used for the different regions, different reasons. So this is the computational methods of the tool. So we can see the FastQC hotspot to find out the what is the quality check, followed by Picard, and for Trimomatic for just trimming down your adapters and all those regions. So then you go to bowtie to BWA high set to star for aligning, and then peak calling. So here you can see the max2. So these are those tools, some of those tools, and then you will do the footprint detection. This is again a new thing. So based on the basis of our requirement and our objective, the analysis steps generally differs with some one or two extra tools. And then these are the motive and all those integrated tools, and these are some of those integrated as I have shown, like Droma Plus, which you can try for the ChIP-seq. So these are some of those integrated tools and pipelines which can help you in understanding. This is the whole pipeline with all those steps that we mentioned, without using for each tool for each kind of step. You can use this kind of integrated and tools and pipelines for full analysis. So now another concept is our DNA methylation. So you know that So this one thing, yeah, if someone wants to say something.
No, Ankit, go ahead.
Okay. So this is now another concept from the epigenomics is the DNA methylation. So here what happens is that there is a covalent transfer of the methyl group to the C5 position of the cytosine ring of DNA by the DNA methyl transferases. So DNA methyl transferases helps us to DNA methyl transferase. This is a C5 position. So mostly to protect the particular DNAs from DNAs and all those. So this methylation kind of is being methylation is one of the phenomena that happens in the DNA. So the cytosine gets methylated, and this CH3 remains attached. So now to understand that where the methylation has happened and how we can identify those methylation. So then we perform, this is the concept of the whole genome bisulfite sequencing. So first we have tried to understand the protein. So let us, if we just go by step. So first we have tried to understand that how which the proteins and is interacting with which part of our DNA as part of before regulation. So all those things that I mentioned, like the copy number abundances or all those with the protein correlations and all. So all those things that we look at in a linear scale is always not linear, and it goes through different steps. So in this process, so we are trying to find out that how it is being regulated before going at the proteome level stage. So here first we are trying to identify that the epigenomic level by the protein and the DNA interactions. Then we looked at the nucleosome disrupted regions, which is and the DNS1 hypersensitive sites, which are helpful in the gene activation. And then we are now looking at the another phenomenon of the DNA is the DNA methylation is by using one concept of the whole genome bisulfite sequencing. So here, as I mentioned, that the cytosine remains for methylated. So when the bisulfite is added to the mixture, so bisulfite, what it does is that it lets the cytosine gets converted to uridine, which in the reads is being taken as used as thymidine. So this conversion can only happen when the sequences or the cytosine is not methylated. If it is methylated, then bisulfite cannot act because it is and it remains in the sequences. It remains in the as cytosine only. When you will go for the amplification, when we will go for the sequencing, so it will remain as cytosine only. So if we just map this one with the known with the genome, then we can easily find out that whenever cytosine was there, thymidine, if it has been converted to thymidine, then it was not methylated or non-methyl unmethylated site. And if it remains cytosine after sequencing, then that means that it was methylated. So we can understand that which regions of the DNA has been methylated using this whole genome bisulfite sequencing. So this can be easily determined by understanding by comparing our treated and the untreated sequences. So this was one of those regions. This was one of those sequencing methods to understand the DNA methylation static whole genome bisulfite sequencing. Another technique which is used is the reduced representation bisulfite sequencing. So what is this? So this is also we are trying to use bisulfite to understand the conversion of the cytosine to uracil, so that we can understand that which region of the DNA. But here is that it enriches the CPG rich genomic regions. So if we wants to understand that in the CG rich genomic regions, how much is the effect of the bisulfite sequence of the methylation in the CG rich island, CPG island region or the CG GC rich regions. So how here we are using we make use of this MSP1 restriction enzyme, so which cuts the DNA at all CCGC sites. So there we had, if you just think, there we had used of the DNase one sites to and understand those regions, nucleosome disrupted regions. So here in our reduced representation, so that's why the name. So from the name only we can understand that though it is the bisulfite sequencing, but here we are not going for the whole genome bisulfite, rather reduced representation. So in a reduced manner of the representation of the particular genome by making use of this MSP1 restriction enzyme, which is cutting the DNA at all our CCGC sites. This enables the measurement of the DNA methylation levels at 5 to 10% of all CpG sites in the mammalian genome. So whatever CpG sites are there in the whole mammalian genome, it helps us to understand those methylation at around 5 to 10% of the methylation level. Okay, so this is by after the concept remains the same, just we are using one more restriction enzyme that is cutting the DNA at all CCGC sites, and then we are treating it with the bisulfite treatment. So that's why it is the reduced representation of the mammalian genome that it has cut, and then finally we are going for and identifying those particular regions. So this is the same, for having the same concept with enriching the CpG rich genomic regions. So now another thing is to, if we try to understand how the protein complexes and all those, what is the structure of those, and to get a whole 3D architecture of the genome. So this till now what we had done is that we had tried to look at a particular fragment of the DNA, particular sharing for a particular protein, how that particular protein or the modifications of the protein is linked with which part of the DNA. And what are the cross links are there between the DNA protein and the DNA in the whole mechanism of those folding. So this is the technique Hi-C, 3C-sec, and chromatin interactions, as well as there is 5C methods as well. So firstly, DNA protein complexes are cross-linked with formaldehyde only. So as I mentioned in the chip-sec also, if you just remember this chip-sec also, the DNA protein complexes are cross-linked with formaldehyde to get the strong interactions. Then fragmentation of the samples. So here cross-link and isolate. Then the DNA is extracted, ligated, and then digested with the restriction enzyme. Okay, so so what is that? The Hi-C detects your chromatin interactions both within and between by covalently cross-linking your protein DNA, and then there is no pull down like in the chip-sec. What we did in the chromatin immunoprecipitation. So whenever this word you can understand this terminology, that immunoprecipitation means there is a precipitation by using the antibody to pull down for a particular protein. But here they are doing it by biotin interactions. They are trying to pull down the cross-linking. We are trying to maintain the cross-linkings and all those between the DNA and the protein regions, as well as the DNA and the DNA regions, to get the understanding that which part of the DNA protein is cross-linked with another region of the DNA protein, to get the structural view of how the nucleosomes are formed, and that how it will open and how those protein will be, or how this translation will happen. So this detects your chromatin interactions from within and between chromosomes by covalently cross-linking protein DNA complexes and it genome-wide interaction maps. So it provides a unique and powerful tool to study nuclear and organization chromosome structure. So Hi-C methods helps us to get all those cross-links and the architecture in a very good manner. So these are the just steps: just digest and biotin fill in, ligation, and the DNA isolation. So whatever protein DNA is, since we are adding the formaldehyde, so it gets the covalently cross-link, so it can be easily pulled, and there will be no disruption between the protein and the DNA interactions. So this was all of to understand the epigenetics, the regions, that what are the different factors, the different protein interactions, DNA methylations, the nucleosome structures. So if anyone has any question regarding all those? So whole genome bisulfite sequencing, reduced representation bisulfite sequencing. No, right? So now moving on. So we
Sir,
have
HI and that chromatin immunoprecipitations are same. So, like, how are they used differently?
now chromatin immunoprecipitation is different in a way that you know that which for a particular protein, like for a particular histone modification, you are pulling down. You want to check for that only, that particular histone H3K, for instance, for that histone which is attached with which part of the DNA. For this, what you will do is that you are using an antibody that you are pulling down that specific protein out of all those crosslinks, and you are just sequencing that fragment of the DNA only. But in high-seq, it's not, you are not trying to pull down any particular protein, rather you are looking at the whole protein DNA interactions. Okay,
Okay,
am I
sir,
clear?
understood.
Yeah.
Sir, can you please go to the slide where whole genome is written?
Sorry.
Sir, whole genome process. I have a question on that.
Is this one or?
Yes, sir. This one.
Yeah. Yeah.
Sir, you told that when the C is not methylated, then it is reacted with bisulfate and make the changes from C to U. Then
Yes.
how it is how it is changed from U to T?
For T, it is done on the readings. When the readings is done sequence, then the base calls are taken as T only. Okay. Okay. So if there is no more questions, so we will move into the another important phenotype.
Yes, sir, it's okay. I was unable to unmute myself.
Yeah. Yes. If you can mute yourself, it would be good. Okay. So another phenomenon is that genotype versus phenotype. Yeah. So can everyone please mute yourself?
Let me see Ankit, who is on mute.
Yeah, so if we look at this is another interesting thing to understand what is genotype versus phenotype. So what is genotype? So in genotype, we know that it is the genetic makeup or the genetic code of the individual, which is your ATGC, the sequences and all. So this is all the information that is found inside the individual cells. right? So now the another thing is the phenotype. So phenotype is the expression of the genotype that is visible for other people and can be observed. So that this we all know from our Mendelian genetics and all the concepts of the genotypes and the phenotypes, that how does genetic codes that is in the form of the DNA, then in the chromosome, then in the nucleosome, and then finally it is being seen in the phenotype, that it is your white hair, red eye, black eye, or the tree, the color of the flower and everything. So this is an important association. But now the question that have always been that what is the association of the genotype and the phenotype, or how much it is associated, or how to understand its association. So there are different concepts to understand. So there are tests which help us to determine whether a genotype is associated with a particular phenotype or not, right? So this is what the question. The question is that whether genotype is associated with a particular phenotype or not, and then we form our hypothesis based on it. This is our null hypothesis, and the alternative hypothesis that there is no association or there is association. So this is how the testing the hypothesis. So here one thing which is one test which is very much important and very much interesting as well. So this is one of the tests has been Fisher's exact test. That is the, I don't know if you all know this famous problem of the lady testing the tea problem. So here the question was that the tea, if you can say by testing. So the lady claimed that she can say that the milk has been poured before adding the tea or after adding the tea. So the question is that whether she has superpower or not. So do you have those superpower to say that the tea, by just testing the tea, that whether the milk was poured before or after? So this is the important. This is the question that needs to be answered. So whether you will uh, whether you will accept the claim that she has superpower or not. So that's from there Fisher gave this formula of this test with the problem, with the answer of this problem of the lady testing the tea problem. So here you can see that or there is another example. You can that if you take out one ball by ball, there are four balls of green and two red, and if you bring out, that will be either green or red. So if you say that I will bring out two green, so what is the probability that it is by chance? So the question here in the statistics is always that what is the probability that is been happening by chance or by guess. So that whether she is saying that it is the milk is poured before the tea, that is by guess, and if it comes correct 4 times, then what is the probability that so what is the probability that she is guessing? So that is the question. So from here, when we calculate, if you calculate the probability, so the rejecting the null hypothesis, that so the null hypothesis here is that she is guessing every time, and the alternative hypothesis is that she is not guessing. So rejecting the null hypothesis, p value is coming is 0.24. So from here, as I think you most of you know that the p value we cannot, it needs to be at least less than 0.05 or lower than that, or in the at the end region to reject the null hypothesis. So here you cannot reject the null hypothesis that she has no magical power. This is same, but just by guessing, one can say that it is one time you can be correct, but that cannot be significant. Okay, so this is how the Fisher's exact test can help us in understanding that whether this genetic type construct or the association that is being made is associated with that particular disease. So this Fisher's exact test and another test, that is our chi-square test. This is sets are your ordinal and it is binary. So remember these two terms. When will you use chi-square test and the Fisher's exact test? When your data type is ordinal and it is binomial, binary. Sorry, it is binary. That is, you have only two options. Like that in that lady testing the tea problem, the question was whether the milk was tested before, whether milk was poured before the tea or after the tea. So there is only two answers, before or after. And similarly was the in the balls with the red or the green balls. So there was also only two answers where you can find out that whether it is only the red ball or the green ball. So here also the question is that whether genotype is associated or not. So it is associated or not associated. So this is the thing. So now when you will use Fisher exact test and when you will use chi-square test. So when the distribution is not hypergeometric, that is what is the meaning of this word hypergeometric? That is, it is not fixed. So for that instance of the T problem also, your answers and your choices were fixed. Similarly, using the balls, their answers and the number of turns is fixed. But in here, let's like understand that here in this question, if we imagine we have 250 individuals where some of them have a given disease and the rest do not. So you randomly pick 250 individuals, and for disease X, there are some people who are disease X and some are not. And among those, you are observing that the 20% of the individuals that are homozygous for this minor allele have disease compared to 10% of the rest. So if you now pick again these 250 individuals, then what is the chance that it would follow the same pattern? So here your numbers, your options are not limited or not same. So this data is not hypergeometric. Okay, so the data is not hypergeometric. So then you try to use the chi-square test functions to understand that whether it will follow the same pattern as it is observed or not. So this is where you will try to put the chi-square test. So chi-square test is nothing, just to find out the differences between the observed and the expected values, and using the so why do you see chi-square test? As I mentioned, so if your data firstly, but you have to try to understand that your data follows a well-known theoretical probability like the normal or Poisson distribution, and it also helps you to trade goodness of fit. Okay, and it is also is a independent statistical test which whether two sets of variables. So here say in the terms that whether the two sets of variables are likely to be related with each other or not. So you have one observation whether another observation will be likely related to each other or not. So when these numbers are not fixed or not same, then you will be using chi-square test. Okay, so this, and also it is used on when the number of relatively large sample size and independence of observation. So this is another important criteria that you need to keep in mind before applying chi-square test, that you need to understand that it is independence of observations, that are the observations are independent to each other. That means that the population you are picking is not going to come up with another population, means another, it is independent all the observations that you made. So this is the chi-square test. So now if, for example, if we try to understand in the context of our genotype and a particular phenotype, so these are the some of those models, and what is and how you can say that, for example, this model is our recessive set model. So here what is said that the mutation in at least one variant on each of the two haplotypes. So this is an interaction between the variants. So there is an uh, so there at least one variant on each of the two haplotypes. So there is an interaction. So recessive set model says that either of the haplotypes you will get one variant and then only disease will occur. So just now understand the link here is that in the recessive set model, what we are saying that mutation is at least one variant on each of the two haplotypes, and then hence there is an interaction between the two haplotypes, means that the mutation in one of those particular haplotype is influencing the other one, though it is normal, but there is the influence, the interaction. So this is leading to the disease. Okay, so this is the recessive set disease. So according to this model, if though it is in the recessive allele, as if something is difficult, you won't get that particular disease or particular trait in that matter. But in this recessive set, according to this model, if any one haplotype is being there in the, if it happens in any of those haplotype, it will interact and it will influence the other one, and it will, disease will be associated. Another is your recessive, that is mutation on both the haplotypes and the variants, and all the variants act independently. And then additive, there at least one mutation at the variant, and the homozygote mutant has twice the disease risk of the heterozygote. Means if the homozygous allele haplotypes have both the mutations, then the effect is twice. And if it is one, then it is, so it gives us the additive domain. And but all variants act independently, means both these has one and both have make twice effect. So these acts independently but are at the uh, but at having an additive effect. Here it is same, but it is a this act independently in the recessive. Here it is that influencing each other. Okay, and this is not independent. And final is the dominant one, that is one mutation in the variant. So the heterozygote and the homozygote have the same disease risk. All variants act independently. So all these three acts independently, but here doesn't act independently. So this is one of those models of the associations of genotype.
Sir, difference between additive and dominant?
Dominant is that when if the variant is present in between in both the haplotype, then also the effect is not the effect is same whether the variant variation is within one haplotype or in both the haplotypes. The phenotypic disease or phenotypic having is same. But in the additive, it happens is that if it is the mutation is in both the haplotypes, then the effect will be much more, means it will get added of what is happens in the one haplotype. Okay, so these are the models. Hope it's clear, right?
Yes, sir.
Okay. So this is an example of the recessive dominant model. So here you can see this is a codominant model, dominant, recessive, over dominant, and a additive model. So here are the control case. So all these models, as I have mentioned, that can it is a control or the case, the control or the disease. So here how it is calculating, then after the statistics, we will calculate the p-value. So finally, what we do is that ACI information criteria, AIC, is generally uh, calculated. So what happens is that it is a mathematical model or mathematical method for evaluating how well a model fits the data. So for example, so now you have these particular genotypes, you have those status that SNP, this is the SNPs. So CC, CG, GG, GCC. So these are SNPs. Now the question is this, the association of this SNP with the disease is fits into which model? Okay, codominant model, dominant model. So how will you understand that this SNP association is how well suited with that particular disease? So how, which model is it the dominant? Is it the codominant? It is the recessive, which is over dominant. So what you will do is that you will collect the data. So for codominant, you will calculate that both the CC, the control and case is how much, and then the dominant, how much is the control and the case for the each of these SNPs, and then you will calculate the p-values, and then you will go for the AIC calculation. So once you find out the AIC calculation, then it helps us in understanding that how well our data is fit. So on the basis of the AIC calculation, we only can say that this model fits this this SNP association with the particular disease is based on this kind of model. Okay, so which if this comes, or if you whenever if you try to do this kind of thing, if so, then you need to calculate this AIC to find out that which model your SNP is getting fitted well. So now another kind of test is the uh, question Armitage trend test. So this is a kind of a test when you try to find out that whether the response uh, is linear or not. So for example, this kind of is in very much use for if you want to add some drugs on a particular disease condition, and you are increasing the doses of the particular drugs in your cells or in your mice or in a clinical trials of the human, and trying to find out that whether your effect of those particular drug is increasing with your increasing concentration or not. So whether it is following a linear trend or not. When the response is binary, that you're either it is responding or non-responding. So this test is used. So this is also your independent testing, that not cured or cured. So this is one example. So between increased dosage and the effect of the drug or the treatment. So here you need to understand, and the dosage levels are your ordinal. So you will follow this Gaussian Armitage trend test to find out that whether it is actually it is giving you a linear trend or not. So this is the trend test for finding. So another thing is that is also which uses your in a different manner. So it also helps you to understand the additive version of the test is mostly used. Okay, so it is also helps in to understand the so it generally considers that the effects are generally additives. For example, when you try to find out the counts of the genotypes, so it gives you the weightage. So in order to measure the effect of genotype I and to detect particular types of enzymes, so weightage WI is given, which for which particular gene it provides and represents the additive effect of those particular allele. So it also considers find out that what is the trend of those data that you are finding. So this was the different tests so more to so the first test there is a Fisher's exact test. So when you will do the Fisher's exact test, when the data follows a hypergeometric distribution, the data first criteria is the data is ordinal, that the responses are binary. Then you will go for the Fisher's exact test and try to find out that whether it is by guesses or by chance, or this SNP's association with disease is actually significant or it is by chance. Then you will follow the Fisher's exact test. Then is the chi-square test, where you will find out your population is following the same pattern as you have observed or not. Then is your uh, as models, your recessive dominant models, and AIC calculation will help you to understand that your SNP and the disease association model is fitting with which well with which data, the mathematical method by calculating those. And finally, question Armitage test is to find out that whether your the thing that you are doing is in linear trend or not, is following a linear trend or not. So for example, in the drug dosage and all those effects, as well as the genetic variations. So if this is also one example, like if the for a particular allele of those variants, if there are genetic variations are causing you the particular diseases or not, that what is the effect of those in those diseases by and it calculates the weightage and provides you in an additive effect. So after this, the another thing is the genome-wide association studies. So I think you might have heard about this term GWAS, genome-wide association studies. So what is this? So this is the studies to identify that the how the, for example, here if you see that the individuals with disease, individuals without a disease, and with a chip you can genotype 5 million SNPs. So this is SNP1, SNP1. So here it is no association with disease. So individuals with the disease and individuals without disease, so these SNPs have no effect. This is the SNP2. So this is also no association to the disease. But this SNP3 is associated with the disease for a particular set of individuals, but it is not affecting a particular set of individuals. Why? How did you understood that this SNP1, if you look at this picture, this SNP1 is randomly distributed among the individuals. It is also randomly distributed among the individuals, but it is not causing any disease. The second SNP is also randomly distributed. It is not causing. And here SNP3, it is the disease causing SNP is associated, as well as in these individuals you can find that these SNPs is maximum in the disease population. These SNP is maximum found, which shows that there is some association for this particular SNP with the disease, as it is found in majority of those individuals, and it is also found in some of the minor individuals in the no disease group. But here it is causing no effect, but here it is causing effect. So this leads to the understanding that why this kind of association is different in different individuals, and how those can be studied and understood to find out the effect of those diseases on a particular trait. And I think it is quite evident from now, and you all are very much familiar this during COVID times. It has been seen that some population has got affected much, and some population it did not cause much effect. If we take out the environment and other comorbidities and other factors, still this concept remained valid. So this is the international HapMap project, which began in 2001 to develop a haplotype map of the human genome that describes the common patterns of the human DNA sequence variation. So this became an important tool for the researchers to use, find genes that affect health, disease, and response to drugs and environmental factors. So what are those genes that are most sensitive to and that affect your health, disease? So all HapMaps you can find out using this is the DBSNP. I think if I have mentioned previously that the database of the single 1000 exomes project and the DBSNPs are those which consists of mostly the SNPs that are present in the human genome. So this is how the HapMap project was launched. So this is the paper, so you all can go through it, that how the international HapMap project was launched to find out those associations. So what is this genome-wide association studies? So one thing is that I think you all have heard of the Manhattan plot. So what are those? That the p-values of the entire GWS, the genome-wide association on the genomic scale. So here, what is in the x-axis? X-axis is your chromosome genomic position by chromosome. Now you will calculate that each particular SNP is located in those particular genomes. That for if you look for a particular disease, now if you want to find out that what is the genome-wide association for diabetes or for hypertension, so you would go and look at each particular genome region, the SNPs on the individuals, and then what you will do is that you will try to understand the p-values by calculating all the statistical tests that we just discussed. So you will try to find out the p-values, and then you will do take a minus log 10 of the p-values, and you will just plot those along with the in the x-axis what is your chromosome genomic position by the chromosome, and in y-axis you will dot the p-values. Now what happens is that there if particular region, for example, for this particular study, for this particular kind of diseases, in this region of the genomic, there is a huge association, and those p-values are very much evident, and those p-values sometimes reaches so high. So this gives the pattern similar to what is present in the Manhattan skyline. Okay, so I think you all can resemble these plots, this plot and this picture. So that's where from the name came for the Manhattan plots. So this is nothing but just the p-values plotted against those positions on the chromosome, which gives us the maximum. Uh, so if it is maximum associated with it, it will have a very low p-value and a very high minus log 10 p-value, and this how it remains, and we can identify for a particular disease which portions of the genome region in the population is affecting much, or which part of the genome is much susceptible to environmental factors or other factors. So this is one of the plots for visualizing those trends or traits. Second is the quantile
Ankit, I think
plots.
we have a question.
Yeah, yeah,
We have a question from Nima Joshi.
yeah,
Am I audible?
yeah, yeah.
So I wanted to ask that when we are taking all of these SNPs and we are
Mhm.
plotting it on a Manhattan plot, how do we decide the threshold, like in the p-values?
No threshold is decided. Generally, the majority of those you can see that are within this p-values or well below lower the p-values, but there are certain which gets above. It's quite high, and from there you can understand the which is more your significant, and it is well beyond your normal what is seen in the other parts of the region of the genome. And definitely you will get the p-values, you will calculate according to the statistical patterns.
Okay, so there is no fixed threshold for this.
So the significance level is one thing, and this you can see that getting high and high, well beyond. So these particular two regions are
Yes, so
taken.
I just had one doubt that above what p-value will be considered that this particular SNP is significant.
That is according to your particular p-value, what you will calculate according to the statistical means. I am saying in the plots, it will be evident from the plots, the particular regions which is showing maximum lowest p-values.
Thanks.
Yeah, so another plot which is done is to measure is the quantile plots or the QQ plots. It is the graphical technique for determining if two data sets come from a populations with a common distribution. So now if you have a population X and population Y, if you are considering two different parts of the region of the world and you are trying to find out that whether the distribution is common or not, then you will use this kind of plots to say that whether it is coming or not. So this is the quantiles of the first data set against the quantiles of the second data set, and if the two data sets come from population whose distributions differ, uh, the point should lie in a straight line. So you will with this 45-degree straight line, you will try to understand that okay, so that the populations, if both are coming from the same distribution from their association, then you will find out that these dots lie in the same place. But if it is not, then you can understand that the two data sets are coming from as a different distribution. So this again gives us an important understand that why particular thing or particular environmental factor can affect differently in two different regions of the world or two different population sets. So this plots helps us in this method helps us in understanding that as well. So this is for the genome wide association studies. Now there is another concept that is your expression quantitative trait loci or EQTL. Okay, so I think you must have heard the word QTL, quantitative trait loci or EQTL. Now here the concept comes that if the particular loci or the gene is mutated, that how much part of that explains a fraction of that, how much fraction of that particular gene which is been changed or is the effect is seen in the phenotype, that how much fraction of that. So in that we from all this we came to know that for a particular disease this loci has a particular different, or this particular loci is causing the disease. Now we need to understand that what is the fraction of that is leading to that expression phenotype. So direct association between markers of with gene expression levels. So what is that expression levels with the variation in the genetic variation. So this is how QTL comes, or quantitative trait loci. So another concept is the cis and trans EQTL. So what is cis and trans? That when the SNP, when it is for example you have a gene and the SNP is distance is less than 1 megabase pair, and when you are getting a particular effect or variation in the gene expression, then you will call those cis quantitative trait quantitative trait loci. But if the distance is greater than 1 megabase pair, then you will call that it is very far away from the distance, then you will call it as a trans. So this is your cis and your trans. So now how you will get an overview of both GWS and EQTL. So we have understood the, firstly we have so here the concept, all the three are integrated. So now again whatever SNPs or whatever we have seen and whatever traits we are seeing, that we are saying that the genotype phenotype trait, but here comes in intermediate steps that we have tried to study using the epigenetics and the epigenomic characteristics, so like the DNA methylation level, protein DNA interactions and all. So what is that particular effect on the methylation level due to a particular SNP is your methylation quantitative trait loci. So this gives us an effect. Now this will further leads in the difference in the transcription level. So this gives us the QTL with that the SNP, the link of the SNP at the transcript level or the mRNA level, that is your quantitative trait loci. Now this particular SNP is how much being seen in the population or in the effect using the GWS, that gives us the trait. So from this SNP going via these intermediate steps of those methylation and all those linkages, protein DNA associations, then it goes finally to the transcription, giving the gene expression. So that how the genetic variation is linked with the genetic expression, how the genetic variation is linked with the methylation pattern, and how this finally is seen in a trait or how it is being affected. So this is how all the workflow it is integrated. So I hope it is the concept is much clearer, right? So do you have any question? Let me check. One question is there, I guess. So what is your question, Hari, if you can say? Okay, so has anyone has any question, or right? So this is the whole concept. Yeah. Yeah. Okay, so this is how the whole traits and all, we can get now a little bit of understanding of how this
Not audible.
not audible.
No, it's audible to me, Ankit.
Okay. Audible, right?
I think you can go ahead. I think
Yeah.
it's audible to me. Yeah.
Okay. Yeah. So this is how the whole system or the epigenomics little bit concepts, which gives us for a particular trait going via these steps. So that's why so this again gives us to appreciate that how these two concepts to use these powerful techniques can lead us to understanding of much more in the particular disease associations of all those. So now we will be looking at some of the resources that are present widely available. So first, this like the ENCODE portal helps you in identifying the different diseases and the different data sets publicly available. Similarly, TCGA was done to identify all the sequences for the pan cancer studies. Similarly, CPTAC was established to find out this consortium to collect all those proteogenomic data for to find more insights on the diseases like cancer and all. So this is one portal, that is our CBio portal, using CBio cancer genomics, which helps in visualizing the TCGA data so that one can, so this is an open platform, so it through which you can explore the multidimensional cancer genomics data. So if you all can open also now only and can look at it, so let us try to go to the CBio portal. So how to explore this portal? Yeah, so I think this page will be open. So if you will just go and search, so this is the CBio portal. Just search in the Google, you will find out, or you can type the http://cbioportal.org. So now we will try to see how we can explore this. So you can see that all the cancer studies, pan cancer studies, pediatric cancer studies, and all cell lines and everything are listed here. And here also the different studies, how many samples have been utilized and everything. So now if we go by this, so let us try to find out all the studies related to breast cancer. So we will click those, and then we can get that how many studies are related to breast cancer, right? So if we do this exercise, so we can see that listed all studies. So it is around 25 studies we can find out for breast cancer itself. So now if we go, then we will select one particular study. Let us select any one study. So we'll let not take some more samples. Yeah. So if we just taken this, and then we can, there are two options you can see. One is your query by gene, and one is explore selected studies. So why these two options? So if you want, if you are not aware of what particular gene you want to look at, and either you want to look at whole data sets, that how it is different and how it is coming in your particular cancer sets, or the thing that you are interested in, so you can go and search through it. So now for us, let us look at that query by gene. So here what you can do all is that, well, without going through all the steps of data analysis and all, you can go and visualize all these things. So I'd recommend that you all go and try to find out those important genes associated with this disease and try to explore as much as you want. For now, here what we can do is that we can just query by gene, and we will query these three genes. So BRCA1, BRCA2, is known for the mutations in the breast cancer, and IDH1 we are is one of the known mutations for a brain tumor. So we will try to see that how those IDH1 gene. One second. So how this IDH1 gene is also coming in the breast cancer. So we will just write these three. Okay, and then we'll submit the query. Okay, so here we can find out the missense mutations. So you can see that it is in the 7%, 10%, and it is just in the 1. And where it's the mutation, Y to C, Y 42 positions has been to C, and everything is you can find out. Samples per patient. And then the mutations. So then you can find out all those that in neither A nor B and everything, the mutual exclusivity, the mutations, missense driver mutations, and everything, that how much it is associated, where is the protein change. So if you just go on and explore along with that in the cosmic and the allele frequency, how many mutations are there in the sample. So all these things you can find out for each disease, the missense, the truncating, the inflame, the splice, the fusions, or whatever. So you can try to find out and explore here. So then we can find out that the in the summary, if you go and you can write the cancer type, and then you can give the so 1 second here we can so you can adjust your minimum cancer. So here is your mutation data and is your CNA data. So you can go for first is your BRCA1, then your BRCA2, and then IDH1. So here just see that your for IDH1 it is giving very much less percentage of alteration frequencies. So from here you can understand that the genes and the mutations in between those two different kinds of cancers are quite different. So I'll just recommend you all to go and explore it. Okay, in a and try to play with this with the different parameters and the different genes and different studies. You could get an idea that how this particular genes and all, and from the TCGA data you can visualize certain diseases and all. So this is if you try to plot, so you can plot also the copy number for each values for the BRCA1. So how many are mutated, no mutated, not. So you can visualize it in a very well manner regarding those data sets. So now you can try to find out the check for the mutual exclusivity for those three different genes, which one so find out the significant gene mutations. So across the different studies you can try to find out. So this is one of the very good resource for the exploring the genomics based studies for the TCGA related. So you all can check other data sets as well with all those data sets available, and you can try to find out the different genes for different diseases of whatever suits your interest to, or if you are working in particular disease you can definitely try out to find out those. So among these another database is there like brain base. So can find out, which also gives us disease curation. So if you just so here also you will find the disease and the associations. So mostly they have provided the associations with the glioma. So what are the genes and what is the type? If you go for omics signature or the omics profiles, if you go for the genome, transcriptome, epigenome, so you can find out. So here you can see the gene symbol is at epigenome, the glioma versus normal. What is the EGFR promoter region? What is the different other data sets? How it is looking, and how in the promoter data sets. This also helps you to give a visualization of a particular disease or data sets, how it looks on. So this is another curated knowledge base for brain disease, but mostly for the glioma and its visualization. So here from here, so like the CBio portal helps you in finding out the uh, all the TCGA related data sets and all those findings. So here also from the brain base you can find out some of those signatures for a particular disease, how it is being associated with the disease and the normal, especially focusing on the glioma and those brain tumors. Similarly, so I'd like to say about this is another portal, BrainProt from the Proteomics lab, IIT Bombay itself. So we here also you can try to find out and help visualize the different brain diseases, both neurodegenerative and the tumor-related diseases, and can try to find out the signatures and can visualize that how it is for a particular gene or a protein in a control and the tumor states. So how it looks different. So these are all you can visualize and can get and without going, all these resources are available to you for you to explore and before if you go for analysis, and if you get a new data, of course after analyzing you can try to visualize accordingly. So now let us try to look at some of the quality check of the raw data. As I said that for any sequencing based study, we need to find out that how the raw data are of the sequence quality. So as in the chip-seq analysis also today I mentioned that there is a huge parameter that needs to be followed before going for further analysis, as well as in the any whole genome sequence data and whole exome sequence data, we need to find out what are the different parameters that are present. So without the parameters, you cannot find out and go for further analysis. So if we go here, so this is a FastQC. So how does it FastQC data generally it looks is that it gives us, so FastQC is most kind of a very popular tool that is for used for all any sequencing kind of data. You will go to the FastQC and you will try to find out what are the basic statistics and per base sequence quality. So the coverage and also the base calls that is being done during the sequencing. So how much accuracy or how much confidence that particular base is called, you can try to find out through the FastQC. So let us look at in how we can perform in the before we go on for the session. So here is there one DNA sequence. So I had kept on installed. So this is the real data which I am showing. So I have kept on the data sequences. So after the sequences, after installing the FastQC, so I have taken the extracted the FastQ files, and then what I did is that find out. So as I was mentioning, so in the real data, you can see that the quality scores are all are on the Q30, and here also you will get a green sign or the green signal showing that your sequences are of high quality. So for using this tool, you need mostly Java, and one another thing you would need is the Linux. It generally in the Unix or Linux operating system only you can use. So that's why I'm just showing you that how it looks the data on a real data sets, not the publicly available data sets. So this is the per sequence quality road. So above average quality should be Q30. So as I mentioned that there are some parameters that we need to look on before understanding that whether we can take it for further analysis. Then is your per base sequence constant. Per sequence GC, so the theoretical distribution should be followed a normal distribution or Poisson should be a Gaussian distribution, but it is not following. But it is okay, as we know that there are some GC content regions more in a particular thing. Then percentage of n sequence length distribution. So all these are gone. Then is the sequence duplication levels. So this is somewhat giving a warning kind of thing that which is not passing everything. Then there are no overrepresented sequences and adapter contents, percentage of adapter contents. So this needs to be further trimmed down when we need to find out the particular this using the multiple samples. So how to get for all the samples in a single space if we try to find out. So just a minute, I will show you. Second. Yeah, so this is another tool, what is the multi-QC. So whenever we have a number of FastQC tools, when number of raw sequencing reads, and we have done all the have done the quality control, then we need to have a multi-QC to see in a single report. So I'll show that how it comes. So the report, if I say that. So this is how your multi-QC report would look like. So here you can see that this is your multi-QC. So here are the number of raw sequences. So whatever percentage of duplicates, percentage of GC and all that we were looking individually at one FastQC. So here we can easily find out everything at a go. Whatever number of data sets are there for you in a particular data set to if you want to go and try to find out in the particular data sets and analyze those, so you can get at a single so multi-QC helps us to visualize all those things in a simple report, so that we do not need to check it individually. So here with the name of the sample wise, you can get the first QC sequence counts, unique counts, duplicate reads, and it helps you to visualize everything. So mean quality scores, you can see that it is above 35, that all the base calls that have been done is done accurately or with a high confidence, and there is no problem with that. Then the sequence quality scores, then the per base sequence content, and then the GC content. So these are the parameters that one looks before going on for the further analysis, that you can visualize of all the data in this simple report. Then the sequence distribution, sequence duplication levels, overrepresented sequence, adapter content, and the status checks that for per base sequence, what is your, which one has passed and which one has not passed. So you can see that this is the parameter where it has not passed, and these are the parameters somewhat has failed for some of the samples, which we should need to look at before going on for further analysis, and without, and all others have passed with some warnings in between. So which this gives a full example of how this data sets we can look on before going for the further analysis. And also we can use different R packages for plotting all this, but generally is a multi-QC is one of those most preferred tool which we can use. Yeah, so this is all for today's session. So hopefully